Hello Year 12, this is the fourth video of the respiration topic 3.3. So we've done the processes of glycolysis, Krebs cycle, and the link reaction and electron transport chain. And now this is a bit of a bitty one about anaerobic respiration, using proteins and fats in the absence of glucose, and when there are inhibitors, a little bit of that, and different things that can happen in respiration and affect the rate of respiration as well. So a little bit about anaerobic respiration is that it's happening in the absence of oxygen. So this would be your GCSE examples of weightlifting, boxing, sprinting, where there is not enough oxygen getting into the body for the amount of energy that's being used at that time. They're fine examples, but also think about things like in animals where you might have a gazelle that sprints and it's life or death situation, and anaerobic is needed then. In childbirth, when mammals are giving birth, there can be a period of time where towards the end especially, the umbilical cord is not able to effectively deliver the oxygen to the baby, but the baby hasn't quite taken its first breath outside yet. So anaerobic respiration can be needed for that. Also, any kind of animal that will have to hold its breath to swim underwater, so like diving fish, diving birds that dive for fish, and anything like that, will have to have some anaerobic respiration taking place in order to do that. So really important process, can't be a long-term thing. It's a short-term fix, and you know as well that that's in absence of oxygen. And you know now that oxygen acts as the final electron acceptor in the electron transport chain. So during the electron transport chain, those protein complexes that are electron carriers in the inner membrane of the mitochondrion, they have to be reduced every time they gain electrons. So every time an electron jumps to them, they are reduced, and they have to pass on those electrons as well to become oxidized again, ready to accept the next set of electrons and become reduced again. So in order for those redox reactions to take place, oxygen acts as the final electron acceptor by taking those electrons away from that last electron carrier so that it can be oxidized and it's ready to accept the next ones. Without oxygen, they all will, one by one, retain their electrons because they're unable to accept the next ones. And then that will mean that the whole process stops, because there won't be a proton gradient, there won't be any chemiosmosis, there won't be the ATP synthetase that is able to produce ATP because of that proton gradient. So that's why we need oxygen. So in the absence of oxygen, you don't have the electron transport chain because it's an aerobic process. The Krebs cycle is indirectly aerobic because of that, because during the electron transport chain, as those NAD and FAD drop off those electrons and they become oxidized, so they are reduced NAD, reduced FAD from early processes, drop off those electrons, become oxidized again, they go back normally to the Krebs cycle, to glycolysis, so that they can be used for the same purpose again. So they go back to oxidize and to collect electrons again. Without the electron transport chain taking place, they are never oxidized again, never regenerated in a way, so you don't get the NAD available for the Krebs cycle to take place. So the Krebs cycle will stop. You could say the same about glycolysis, except that there is a reaction that acts as a mechanism for glycolysis to take place. So what anaerobic respiration is, it's glycolysis taking place over and over and over again, lots of times, so that ATP can be produced in small amounts. Because if you remember, in glycolysis, so you're not going to be able to see this really clearly, but you should know what part I'm talking about, because you need to know the rest before you do this bit. So glucose is phosphorylated in glycolysis, split into triose phosphates or glyceraldehyde 3-phosphates. and then series of reactions, you get pyruvate molecules at the bottom. So what's happening here, remember as well, is that 2 ATP are needed there for activation energy. 4 ATP, 2 on each side, are produced down here, and 2 NAD are reduced. So 1 here and 1 here. So what we get then, we'll just summarise that here from glycolysis. Imagine that there's nothing else taking place. From glycolysis, you get your net gain of 2 ATP from 2 spent, 4 produced. You get 2 reduced NAD or NADH, and there will also be 2 pyruvate molecules. Okay, so you probably know from GCSE that if you do anaerobic respiration for too long, something builds up in your muscles, lactic acid. So this is where we're going with this. What's going to happen is the body uses what we've got excess of here to make what we need for this to just keep going on and on and on. And if it keeps going on for this short period of time, then we'll get all these little ATP. This is nothing really compared to the 38 that we could get with one glucose molecule going through the whole process. Should be worth about 38 ATP, remember. So this isn't great, this is short term, but it's a short term fix that's worth having because there's these small amounts of ATP and this can happen over and over. So in animals, because there's a different thing that happens in plants, in animals we have anaerobic respiration, and what we do is use this pyruvate is going to oxidize the reduced NAD. So for the reduced NAD to become oxidized, to just become NAD again, this pyruvate has to be reduced. It has to take those electrons. So pyruvate and NADH will give us the thing that builds up in our muscles when we're doing too much anaerobic respiration. We get lactic acid or lactate. And now, the main reason for doing this, or the reason for doing this, is that we get that NADH, that reduced NAD has been oxidized, and we get NAD. And that allows the glycolysis to take place because we just need the NAD to come back in here to be reduced again. Because it's not just about reducing that NAD. Obviously that NAD does get reduced, but it must oxidize an intermediate here to allow this process to take place. So it is important. So the pyruvate gets converted to lactate and the NADH gets converted to NAD, and that allows glycolysis to continue. You know what happens if this goes on for too long is that we then have to stop at some point and we have an oxygen debt. So the oxygen debt you have done about briefly in GCSE is paying back the oxygen to break down the lactate and continue. So what happens is the lactate gets broken back down into pyruvate, and then pyruvate can continue through respiration. So it can, some of it gets broken down to pyruvate, some of it gets broken down to something else, but basically the oxygen is needed for that to take place. So you take in oxygen and that happens. Then a different process that we have in plants. So in plants, that's not very good. In plants and also some microorganisms. So some microorganisms. And I'm thinking of the classic one here, which is yeast. So yeast will perform this type of anaerobic respiration. What happens is you start off with the same stuff. So again, we've got in glycolysis, now we've got pyruvate and NADH. Now this is turned into something different. And if you're thinking of beer and wine making or bread making, then that'll help you. So there's a type of alcohol, which is ethanol. And carbon dioxide and NAD, which is what we're looking for. So when you're making beer and wine, then obviously ethanol is what they're trying to make. When you're making bread, the ethanol evaporates and the carbon dioxide is what makes the bread rise from the yeast performing this. So that's the anaerobic respiration. Anaerobic respiration is glycolysis happening repeatedly in the absence of oxygen. The Krebs cycle and the electron transport chain are unable to take place, but for this to keep going, you need NAD to be oxidized again, reduced NAD to be oxidized, and that involves this lactate reaction in animals and this other one in plants and microorganisms. Okay, that's anaerobic respiration. So now we just need to briefly look at, and it's not a lot of detail really, so don't go, if you go reading about this, then try not to get, you can get, read a lot more into it, but don't go too deep and panic because there's not a huge detail needed for this. So this is now about having insufficient glucose. Okay, so if there's not enough glucose available, then we turn to other macronutrients. So the other large molecules that we consume that can give us some energy if we need them. So glucose isn't available, and this is like diets like Atkins and ketone diets and things like that, which scientifically aren't recommended and certainly aren't recommended as a long-term solution because they're not meant to be. Glucose is needed by the body, so you do need carbohydrates. The brain works just off glucose anyway. So when there's not enough glucose available, then we have proteins and fats that are going to be fed into this and then will eventually lead to the electron transport chain and some production of ATP. So if proteins are used, proteins can actually be converted, so broken down first of all into amino acids, and then those amino acids can be converted into acetyl coenzyme A. So here, end of link reaction, start of the Krebs cycle. So if you get a protein, break it down into its amino acids, they can be modified, converted, they obviously have to go through some different reactions, things added and taken away from various amino acids, and you can form acetyl coenzyme A so that the Krebs cycle and then the electron transport chain can continue from that point. Then lipids, they obviously break down into glycerol and fatty acids. Now glycerol can be converted and modified to become this molecule here. So this one, so I've been calling it triose phosphate sometimes, and sometimes glyceraldehyde 3-phosphate, and that's because it is a triose phosphate. It's a 3-carbon sugar with a phosphate added. But glyceraldehyde 3-phosphate, it is similar-ish in structure to glycerol. So glycerol, from the breakdown of lipids, can be converted into glyceraldehyde 3-phosphate, which is this one. So then breakdown of fats can lead to the reaction and the metabolic pathway occurring from that point. Also, when you break down fats, you get glycerol, but you also get fatty acids. Fatty acids, remember, are the long chain molecules, and in those long chain molecules you have remember it's hydrocarbon chains, all the carbons and hydrogens, and they can be snipped up into smaller parts And again, if they are modified, then they can be converted into acetyl coenzyme A. So that's a useful one, is that when you break down the fats, from that point, if you have acetyl coenzyme A, then the Krebs cycle and the electron transport chain can occur as normal. Okay, so that's if there's not enough glucose, proteins and lipids. Break down modifications that you don't need to be able to chemically explain. You just need to know what the modifications are at a very basic level, that they the parts that are then broken down, like the amino acid, like the glycerol, and where they can get fed into that that metabolic pathway. Then other than that, there's just a few sort of general things to think about as well. So the fact that we have a reaction taking place here, and there's going to be something that affects the rate of reaction. So the things that affect the rate of reaction here will be the amount of oxygen available, the amount of glucose that's available, the temperature. Remember, temperature will always affect enzymes and the rate of reaction of the enzymes. And then the other thing for this is the amount of ATP that's needed, because if there's a lot of ATP available, then that can inhibit parts of this. Very clever. So if there is lots of ATP around, then this won't take place as much. When there's less ATP available, then respiration will occur more. So lots of different things affect the rate of reaction, and if we get to do practicals when we go back, then we will see some investigations into the rate of respiration. Then the other thing is the inhibitors and the poisons. So some of you have done this, so you've read a little bit about this in the questions. And there's lots of different ways that inhibitors and poisons can affect what's going on. I'll draw a little diagram. But generally we're talking about the electron transport chain. Okay. So in the electron transport chain, I do a really brief version of it. Okay. In the electron transport chain, you've got the electron carriers that act, some of them as proton pumps, and then I've not drawn here, but we've got the stalked particle that has the ATP synthetase here as well, which will be in that same membrane and associated with this electron transport chain, which is where the ATP is actually produced. So there are different poisons and inhibitors. Now you don't have to necessarily, I don't think they would ever say name an inhibitor. They could ask you to name one or two, but you're not expected to reel off a big long list of inhibitors. I do think though it's useful to know a bit of a list of them, so that if you see a question that is about an inhibitor, you can recognise the name. So for example, if you had one of them is like rotenone. If you saw a question that had the word rotenone in it, it's one of those ones where it would give you the information in the question, but you could pick up on the fact, this is probably about inhibitors, and then you could start talking about all this stuff. Okay, so these are the sorts of questions where they will give you the information and tell you what these things, exactly what each one is doing, and expect you to apply your knowledge of the electron transport chain to that as well. So the sorts of things you could have, cyanide is normally named, amytal, rotenone, carbon monoxide is one of them as well. So the different ones have different things that they do, but basically there's main things that they can cause problems with, and one of them is that this inner membrane here. This, remember, is where we're going to have chemiosmosis. We're going to have hydrogen ions that are pumped this way, and we need a build-up of hydrogen ions up here so that they can flow back through the ATP synthetase and form the ATP. So one of the problems is that some of these inhibitors and poisons punch holes or form channels in that inner membrane, and if there's a channel or that's broken down that inner membrane at all, then it's going to allow protons to leak back through here and then that gradient is not going to be as effective, and it's not going to be reached in the same way, and it causes ATP production to decrease. Another way that it can happen, and this is a common one, is that it can prevent oxidation of an electron carrier. So sometimes we inhibit, or they could inhibit, like cyanide, stop oxygen from accepting the final electron. So obviously, like I said before, that backs up and the whole chain of redox reactions can't occur. But anywhere along there can be inhibitors that can attach to or affect change the working of these electron carriers, and then that will mean that the oxidation is prevented, or it could prevent the reduction of some of those. Okay. So I know that's quite a bit, and it's a few bits and bobs, like I said, but I'll point you in the direction of some sort of summaries of these points, but they're basically not things to go into massive detail about. Each little part, it would be a few bullet points, but it's understanding all of what I've taught you before, the glycolysis, link, Krebs, and the electron transport chain. If you understand them very well, it's learning little bullet points of what I've done today and being able to apply that to everything else and the other processes.